Swayam Prabha. Digital India. Educated India.
In this lecture, we will mainly discuss on protein sequence databases. So, let us refresh ourselves about the last class. So, you remember what did we discuss in the last class? Yeah, this mainly about the protein structures. So, what is the building block for protein structures? Amino acids, right. So, amino acids are classified in two major groups. What are two major groups? Hydrophobic residues and hydrophilic residues, and the hydrophobic residues we subclassified into different groups. What are the subclassifications? Yeah, so you can have aliphatic amino acids, aromatic amino acids, sulfur containing amino acids and glycine. For the case of hydrophilic residues, we classified into positive charge, negative charge and polar, right. So, we have classified based on the amino acid properties. Then we discussed about protein functions. Do you remember what are the different functions we discussed in the last class? Enzymes, antigens, antibodies, structural proteins, regulatory proteins, right, and the sweetener. So, blood clotting proteins, thrombin, so transporters, right, the hemoglobin. So, we discussed about the various proteins with they perform different functions. Then we discussed about the structure function relationship. To understand the functions of proteins, right, the structures will help, and mainly the structure of a protein dictates its function. So, it is very important to understand the structural level. So, if you look into the various organization of protein structures, they are mainly classified into 4 different groups, right, primary structure, secondary structure, tertiary structure and quaternary structure. In addition, there are 2 structures in between them, they are super secondary structures, the combination of different secondary structures plus domains. Let us say in a protein some part of these residues they can fold and they can perform functions, they are called domains. So, what is the primary structure? Right, it is arrangement of different amino acid residues, right. So, primary structure gives the sequential order of these amino acid residues, and secondary structure provides the arrangement of these residues in some specific shape. For example, here this is a kind of spiral shape. So, this is called alpha helix. So, here you can see this is a kind of ladder. So, this is called beta strand, right. So, here the next one is tertiary structure. Tertiary structure will provide the atomic coordinates of all the atoms in each residue. So, you can get the location of each atoms, right. If you take the XYZ coordinates, you will see where is the location of each atom in a residue, right. So, in the XYZ coordinates. Then the assembled subunits of these tertiary structures that provide the quaternary structures, right. So, today we will discuss mainly on protein primary structure, and where shall we get the information from for the protein primary structures, are there databases available and how to extract the data and how to utilize the data, right. So, I discussed earlier primary structure is a specific combination of amino acid residues in a protein, right. Totally how many naturally occurring amino acid residues? 20 amino acid residues, right, 20 amino acid residues, but these 20 amino acid residues can make various combinations, right. You can see the combination of various possibilities, right. So, is it fine to have any possible combinations or only specific combinations are allowed or possible for protein function? So, this is kind like English dictionaries. So, if there are 26 alphabets, right. So, with these 26 alphabets you can make various words, sentences, right, and you can put these articles and books and so on. Likewise, there are 20 different amino acids, right, they can also be used to make several functional proteins. In the case of these 26 alphabets in English, right, so only some specific words are present in a dictionary. For example, if you write like this education, right, this is present in the dictionary, and this university, this is also present in the dictionary, but if you write like this, it has no meaning, like this, right. So, I put UN and all this 4, 5, 6 I's and some V's, right. So, it has no meaning, it is not available in the dictionary. Likewise, the proteins formed by different amino acid residues, but there should be some specific combinations, right. And then again if you see in this chain, right, this is the main chain, so like the varying chain. So, these are the beads. So, this is common for all the amino acids, this is called the main chain, right, you can, this is called this main chain, and here the beads are different, right, this is called the side chains. So, if you see here same bead can repeat again, they can come close to each other or they are far away each other, but nature selects a specific combination of amino acid residues to form a functional protein, right. In this case you can make any polypeptides, but all polypeptides are not proteins, only the specific combination of these polypeptides they form a functional protein. So, I show an example. So, this is the amino acid sequence for human hemoglobin. So, they have the specific combination of amino acid residues, and even if we disturb any of these amino acids, for example, we discussed earlier, right, for the case of, right, the glutamic acid 6-valent, it causes the disease sickle cell anemia. So, likewise if you, even if small changes in a protein that can alter the function of a particular protein, sometimes they may lead to diseases too. The combination of these amino acid residues are very important for a functional protein. So, what are the information we know from the primary structure? So, the primary structure gives the linear sequence of amino acid residues. So, it will give you the specific combination of these residues, that is we know. And the second one, it includes all covalent bonds, because when you make two different amino acids, right. So, what will happen, how to combine the two different amino acids? Covalent, by the elimination of the water molecule we form a bond. What is the name of the bond? Peptide bond, right. So, you have the peptide bond. So, when we know all the covalent bonds between all the amino acids, right, then if you for relative arrangement of these amino acids are not specified, we do not know where they are located. So, we know only the combinations and we know the covalent bonds. So, now there are various sequences available in the literature when they synthesize a protein, right. So, they get these protein sequences, right, they publish the data in the literature. So, now to understand the features, are there any important characteristic features among these sequences, it is essential to have a set of sequences. If you have a collection of sequences, then we can easily know, okay, what is the preference of a specific residues in a protein, right. Are there any bias of any specific amino acids, because if you have 20 different amino acid residues, right, are there any amino acid would prefer to occur more in any proteins or prefer to occur less in a protein, and so on. So, what happened then, the Margaret Dayoff, right, from the Georgetown University. So, she collected the sequences of various proteins and published a book of atlas of protein sequences. She published very large volumes of books which contain the sequences of proteins, right. Later on they changed this information, they converted the information available in the book in the form of a database, right. This is how they got the database called the protein information resource, right. There are various databases which contain the information regarding protein sequences, like the Munich Information Center for Protein Sequences, right, NCBI protein database, and PIR protein information resource developed by Margaret Dayoff, right, from Georgetown University. In the meantime, the PIR of Protein Research Foundation, they also developed to collect the data, it is from the Osaka University. Then SwissProt, it is the unique resource for the protein sequences, and they created and maintained from Swiss Institute of Bioinformatics. So, they have two types of information, one is the manually curated sequences and also the translated sequences from the DNA sequences, this is called the TR-EMBN. Later on they combine both these major databases like PIR and SwissProt, right, and they formed a consortium called the UniProt, right. This is the universal protein knowledge base which contain the information regarding protein sequences plus other information what we can use as a tools. Although these two major databases PIR and SwissProt, they merge together to share the information regarding protein sequences, they develop tools by these groups, they maintain by themselves. So, you can share and you can get the sequences from UniProt, but if you want to use any of these tools, there you can use the SwissProt or PIR So, they have different specific tools to analyze these protein sequences. So, then I will explain what are the information earlier they started to collect and how they merge together to form the UniProt. Protein information resource, okay, this is PIR, right, this is the one which initially started from Georgetown University, right. So, they collect the data, so they translate the data available in the book in the form of web resource, right, and they classify it into 3 different groups. One is the protein ontology, right, this is PRO, and iPro class is contains integrated protein knowledge base, and the iPro link this contains literature information and knowledge. Essentially, this is the integrated protein information resource for understanding the genomics data, proteomic data as well as systems biology research. So, try to put it together. So, they collected the data and they classified into 3 different categories, and each one presents in a different ways. For example, in the case of protein ontology, mainly what they give, they represent protein objects with the descriptions as well as the relationship, they give the different function information. Here in the iPro class, right, they give the function analysis and the mapping as well as mainly the sequence information. In the link, they give the source for the text mining and as well as the ontology development. You can have the different links with the different aspects like the bibliography, mapping and so on. Okay. Then how to use this database? So, the iPro class they provides value added information on various aspects like protein sequence, protein structures, families, functions, interactions, expressions as well as the modifications, right. So, they give the protein sequence, and for each protein sequence they added information regarding other resources. So, they have various options to search the data. One is the simple search. In this case you can give any keywords and you can get the data using that specific keywords, or they developed advanced search options. Here they have various conditions. So, you can give any of the conditions based on user choice, and the database uses all the information as a query and then provide you the desired output. So, here first this is the simple text. So, you can select a database at the PIRSF or iPro class, because iPro class contains the mainly the sequences. So, you click on this iPro class, right, and here there are various options for the query, whether you want to search with any specific field or you want to search on any field. So, here we use any field. So, then I put human lysozyme, right. So, I want to get the information regarding human lysozyme. So, I click human lysozyme and search, right, you can click on search. So, this will give you the whole picture. So, it will take your query, right. So, last time we discussed about databases, right. So, what is the generally used biological databases? Relational database, right. So, they use the query and they use language, they use structured query language, right, SQL, right, they use structured query language to pick up the data from the database. So, they use this lysozyme as your query any of the fields, right, and wherever it finds the name lysozyme in the data, that will display the entries. So, if you get there are several entries, right, these are the protein ID and this is the protein name, right. So, they give the length of the protein. What is the meaning of 148? 148 amino acid residues in the particular protein, right. So, then the organism, right, where is the main organ species for that particular protein, and this PI or SFID and related sequences, and what are the matched field, why are they matched field, because human lysozyme is the field we gave, it matched the paper title, right. Sometimes they have paper title, right, somewhere you can give the protein name, for here it matched with the protein name, right. So, it matched wherever it matched, so it showed where they found the match, right. So, now if you take this one, so click on this, then you can go get more details on that particular specific protein. So, go with iPro class and they have a link with the different other resources. So, go with the iPro class, so then we will get the information. So, here you first we give the protein name, so then give the all the other synonyms, all these things, right. Then we get taxonomy. For example, this is the from Homo sapiens, from human, right, this is the mammalia, and you can see the lineage is eukaryota from metazoa, right, and go up to the last one, that is Homo sapiens. They give all the lineage of the particular protein. Then they give the gene name and the different keywords they give, right, because it is important to have several keywords, so that one can obtain the desired information with any different searches, right. You can get from the 3D structures or amyloids or the disease mutation, because it contains several disease mutations, disulfide bond and so on. So, you can obtain the specific protein sequences based on various search options. This is why they give various keywords in this particular protein. Then give the function, right. So, this is the mainly a bacteriolytic function, right. So, here the tissues and body fluids are associated with the monotype monophase system. So, they give the specific function of a particular protein. Then you give the cross references, right. So, they give the bibliography, DNA sequence. What are the different DNA sequence databases? DDBJ, EMBL and GenBank, right. So, they give the link with all the databases, right. They give GenBank, EMBL or the DDBJ and so on. So, now give the structures, I will discuss in later classes, right, when we have the 3D structures. So, they give the codes for the protein data bank codes, right. So, they give the codes for the protein data bank. Okay, this is called PDB, right. They give the IDs. Then give the family classification, right, for the based on the folding and other classifications, PFAM domains and so on, right. Then give a sequence. Right. Here this is the sequence of their particular protein, right. So, for the human lysozyme, that so you can see the number of residues, right, in this particular protein. So, now this is the IPRO class you can get from the PIR database. During the same time, the Swiss Institute of Bioinformatics, they also tried to collect the data and they developed database called the SwissProt, right. Later on UniProt comes mainly from these prots because they have the various functional aspects and they collected lot of information regarding protein sequences plus the analysis. So, this SwissProt is established in 1986, right, from the University of Genova and EMBL data library. This is the curated database, right, and the major aspect of UniProt, this is high level of annotation. They give much information regarding a particular protein. I will in the later slides I will explain some of the major aspects of this UniProt. It provides high level of annotation. For example, they give the description of the protein and various functions, right, and the structures or the post translational modifications and different variants, diseases and the interactions and so on. So, give wealth of information for a particular protein. Then second aspect, this is the first aspect. This is a high level of annotation and the second aspect is it has minimum level of redundancy. The redundancy, if you see the sequences, they provide minimal. Sometimes you can get the same information from different resources. They try to put in the resources, but try to minimize the redundant information, right. And the third aspect, it also have high level of integration with other databases. So, they give the links to various other databases in the literature. So, there are three major aspects of this UniProt. What are three major aspects of the UniProt? High level of annotation, minimal level of redundancy as well as high level of integration. So, these are the major aspects. This is the reason why several users for this particular database called the UniProt database. Then they have another supplemented data, this TREMBL, this is translated sequences, right. This is the computer annotated supplement which contains all translations of the EMBL nucleotide entries which are not integrated into SwissProt. Mainly SwissProt, they have manually curate the sequence and they provide very accurate information. In addition, they also provide the computer annotated sequence from the DNA sequences. This is the reason, if you see, it has the this is manually curated data, this is 0.55 million and this 73.7 million, this is the computer annotated sequences and totally we have about 75 million sequences, right. This is a good amount of data, right, of protein sequences. So, you can use this database, UniProt database at the website unipro.org. You can get the all the information regarding a particular protein. So, we look into the UniProt. So, if you want to use a database, as we discussed in the previous classes, if you want to develop a database, right, so you have to maintain the database. It is easy to develop a database, but it is very difficult to maintain a database, right. So, there should be a uniform increase of data and there should be a very reliable data, right, and then very clean data, right, that is very important. So, if you see this UniProt, you can, this is the data for the UniProt SwissProt and this is for the TREMBL, right, this TREMBL is SwissProt, this is the manually curated one, this is the computer annotated one. So, you can see gradual increase of the sequences. They started working on these sequences and several efforts have been put to curate the data. So, you can see gradual increase in data. Likewise, the TREMBL also they can see gradual increase in data, right. Maybe they might have deleted some of these entries, this is the reason there is a slowdown and then again it is growing up. So, they have gradually maintaining the data, continuously maintaining the data and gradually increasing the data. This shows the growth of this database and if you see the citations, you will get the citations also improving every year, because for any protein sequence information one has to use this UniProt database. So, now if you look into the classification, which type of data are dominant in UniProt database, right. If you see this is a classification, so mainly you can see the data dominated with which type of organism, which kingdom of life, this is mainly the bacteria. You can see this is the bacteria data, this is highly dominant, and followed by eukaryota and then virus, right, and then little bit and then about the archaea. So, you can see similar preference for the viruses and the archaea. Right. In both cases, even for the Swissprot entries as well as the TRML entries, you can see similar level of data mainly dominated by bacteria followed by eukaryota and viruses. Now, the question is, there are many proteins in the database, what is the average length of the protein? So, there are many proteins, right. So, there are what is the average length of the protein? So, what is the average length of the protein? 3115 amino acids based on the all the proteins deposited in the UniProt database. Usually, if you look into the sequence length in different proteins in UniProt, so you can see this is mainly this region. This region means that is about 100 to 400 residues, right. Most of the proteins are small like proteins or medium range proteins, around 200 to 300 proteins, right. You can see the higher peak around 250 to 300 proteins, right. And some proteins which are quite long, which is more than 1000 residues, right. When finally if you take the average length, it is about 315 amino acid residues in each protein, but there is most of the proteins they are in this region. So, there is about 100 to 400 residues. So, now if you look into these different proteins, right, are there any bias of different amino acid residues? Totally how many amino acid residues? 20 residues, right, different classifications, polar, non-polar, charged and so on. Are there any bias? Any specific residues are predominantly occurring in protein sequences, right. So, here I show the data. So, this data shows the gradual decrease, and here this is classified based on the different groups, right. If you see, there are some amino acid residues which are highly occurring in protein sequences, right. What residues occur dominantly in protein sequences? Leucine, alanine, glycine, right. So, here these are the residues which occur predominantly in protein sequences. When you look into the rarely occurring amino acids, what are rarely occurring amino acids? So, tryptophan and cysteine. Okay, this data I obtained from few releases ago. This is the current data release one. So, if you see the tryptophan, there is just 1.2%, and for the cysteine the same 1.2%. So, highest occurring if you see lysine is 9.8%, right, and the alanine is the 8.9%. If all the 20 residues are randomly distributed, right, what is the percentage of each amino acid residues? 5%, right. But here if you see that is not like that, right. Some residues are 10, that is double the amount, and some residues are only 1.2, right. So, this is mainly due to the structural and functional aspect. This is very important, right, for to fold a particular protein in the globular shape as well as to maintain the stability and for the functions. With these hydrophobic residues, they tend to form a hydrophobic core that is initiating the protein folding and maintain the stability. Likewise, you can see a sufficient number of charged residues, like for example, lysine and arginine, right. So, here the lysine and arginine. So, what is the percentage of lysine and arginine? Yeah, 5% lysine and arginine 5.6%, as well as the polar residues, that is serine, threonine, you can see sufficient percentage, because these tend to form the electrostatic interactions as well as form the hydrogen bonds. So, now these residues have specific bias in the any protein sequences.